Welcome everyone. Today I will be solving question number 43 of the 2017 GATE Biotechnology paper. Shown below is a plasmid vector P and an insert Q. The insert was cloned into the BAM H1 site of the vector. The recombinant plasmid was isolated and digested with BAM H1 or ZO1. The results from the digestion are shown here. Now the question is that you have to find out that which one of the following explains the digestion results shown. There are four options given to you. Option A is the insert did not ligate to the vector. Option B is one copy of the insert ligated to the vector. Option C is the insert ligated the vector as two tandem copies. And option D is the insert ligated to the vector as two copies but not in tandem. So let's look at each option carefully. So in the first case where no ligation had happened, that means the insert is also free and the vector is also free. There is no recombinant DNA formed. So if you have a digestion with BAM H1, the vector will be linearized and you will get a 5 kb linear DNA fragment. And since there is no digestion happening of the insert, you get a 1 kb fragment. So if you see this image, you do get a 5 kb fragment and a 1 kb fragment. So this could be possible. Now, if you had digested with ZO1, you will not get a linear DNA fragment for the vector because it does not have a ZO1 site. And you will get a 0.3 and a 0.7 kb fragment of the insert. But since that is not observed in this case, option number A is therefore wrong. Option B is that only one copy of the insert is added to the vector. So if this is the recombinant DNA and this is the gene of interest that was added, the ZO site will be here, the BAM site will be here. So if you cut with BAM H1, you will have a 5 kb fragment, which is this, and a 1 kb fragment. Again, you can see that. So this can be the case. But when you cut it with ZO1, you get only one cut here. And this 6 kb fragment or 6 kb recombinant DNA will get linearized and you will get a 6 kb fragment, which you can see here. But it also has a 1 kb fragment, which will not be formed in this case. So that is also not the correct answer. Option C is that two copies of the insert are added and in tandem. So in tandem means that DNA is repeatedly added numerous times in a head-to-tail fashion in the same sequence. So let's take that example. If the DNA or the insert was added in tandem, what would happen is there will be one ZO site here, one ZO site here, a BAM site, and BAM site. So if you cut with BAM, you will get a 5 kb fragment and two 1 kb fragments, which will appear as the same band. So this can also be the case. When you cut it with ZO, what you get is a 5 kb fragment plus, since the cut is here, a 0.3 addition on one side and a 0.7 addition on the other side. So 5 plus 0.3 plus 0.7 will give you a 6 kb fragment. And what will be remaining is again 0.3 plus 0.7, giving you 1 kb. So you observe that. So you get a 6 kb fragment and a 1 kb fragment. So this is one of the correct answers. Now let's see the last option where two copies are added but not in tandem. Means they are not in head-to-tail fashion. So in that case, one copy will have a ZO site here and the other copy will have a ZO site here. So what happens when you cut the DNA with BAM H1 is that you get again a 5 kb fragment and two 1 kb fragments which will appear as the same band. So this can be possible. The second is when you cut it with ZO, that is ZO1 here and here, you will get a 5 kb fragment plus 0.3 on both sides. So 5 plus 0.3 plus 0.3, and you will get a 0.7 plus 0.7 fragment. This fragment. So you should get a 5.6 and 1.4 kb fragment, which is not what you observe here. So option number D is not correct. So amongst all the options that we have seen, there is only one option which is correct, and that is option number C, that the insert ligated to the vector as two tandem copies. That's it for this question. I hope that was easy for you to understand. Thank you so much for listening.